Hi there guys, welcome to another video. Mr. Phillips for year 12 or AS level biology. Today we're moving on to a new module, which is the gas exchange module, and we're going to look at the need to have gas exchange systems in our bodies. So just to start off then, why do we need to breathe? I think that's quite an important question. If you can't answer that using these six words, I think you probably need to press pause and have a think about it. But press pause, see if you can answer it, and then I'll start again. Okay, so what we're going to do, let's have a look at it. Let's think about it. Why do we need to breathe? Well, we need to breathe because we need to be able to complete the process called aerobic respiration, the cellular reaction called aerobic respiration, which releases energy from our food. And obviously that happens in all our cells, and in order to get that oxygen into our cells, because that is needed for respiration, so obviously oxygen plus glucose goes to carbon dioxide, water, and energy. We need to get that oxygen into our bloodstream. And when we do respire, we produce the waste product carbon dioxide, and that obviously becomes carbonic acid in our cells, which lowers the pH, and therefore we need to remove that from our bodies, which we also do via the lungs, through the process called excretion, and that is the removal of waste products. So why do we need to breathe? Because we need to breathe to get that oxygen in, to get that carbon dioxide out, and obviously it's the exchange site of the alveoli that make that possible, because we are too big an organism to be able to do it just through diffusion alone. So starting off then, single-celled organisms are a bit different. They will obviously just be able to use diffusion. Okay, so if you imagine you've got a single-celled organism here, okay, an amoeba or something like that, okay, it's got a relatively large surface area to volume, well, it's got a large surface area to volume ratio, okay, and all of its environment, all of its cytoplasm is close to the environment. So all the oxygen, all the nutrients can just diffuse straight in. It's got a large surface area, it's not large surface area to volume ratio, and any waste products can just diffuse simply out. And oxygen obviously can just diffuse simply in. Carbon dioxide can do simple diffusion out from a higher concentration to a lower concentration. But in multicellular organisms, there are too many layers of cells for diffusion just to take place across. And this obviously is a big issue, because let's imagine your arm or something like that. That's probably got a billion cells in it. So if you imagine that your diffusion may work effectively across maybe one or two cells, then it's going to sort of struggle to get anywhere. So if you just relied on diffusion into your body, imagine that was just a relatively simple organism with a larger surface area to volume ratio, which is a multicellular organism. That distance and those barriers are going to be far too great for diffusion to occur across. And that's going to slow down the rate of diffusion, so it's too slow, or it just doesn't happen at all. Now, we're adapted to diffusion, like all larger organisms are, by having lungs, which allow us to breathe in and out. Obviously, if you're a fish, we're talking about gills, and we have small intestines for the food that we eat that's digested to diffuse across into the bloodstream as well. And they are exchange sites that increase the surface area in our. So this all keeps going back to surface area to volume ratio. Okay, and basically you might need to work out the area, which is doing the base times the height, and then the surface area, which is obviously the area times the number of sides. You might need to do the volume, which is base times height times depth. Okay, and then you do surface area divided by volume to work out surface area. So if we have a look at these examples here, we've got a couple of different cubes. So we start off with a small cube over here on the right hand side. Just lost my cursor, sorry, there we go. So if we look at this one, we've got a surface area of 1 squared, surface area of 1 times 6, or 1 squared times 6. And obviously 1 squared times 6 is 1 times 1 times 6 sides. So that means this side's going to be 1, this side's going to be 1, this side's going to be 1, and the other 3 sides are going to be 1. So it has a surface area of 6. We then have a volume of 1 cubed, because the height, the width, and the base are all the same, so 1 times 1 times 1, and that's going to have therefore a surface area of 1. So then you do 6, the surface, divided by the volume 1. Sorry, I said that wrong, so we have a volume 1, that equals 6. So it's got a relatively large surface area to volume ratio. We have a look at this one. You've got, again, to work out the surface, you've got to do 2 times 2 to work out the surface of one side, which would be there to that, like this side and this side. So 2 times 2, and then you've got to multiply that number by 6, which equals 24. and you've got to do 2 cubed, so 2 times 2 times 2, which equals 8. So that equals 24, which is the surface area, which is obviously bigger because it's a bigger structure, divided by 8, which is again bigger than 1 because it's bigger structure. But when you do 24 divided by 8, the surface area to volume ratio is actually smaller, it's only 3. And then this one, we've got 3 cubes, okay? So 3 squared, it gives the surface area of one side, which is 9, times that by 6 equals 54, and then 3 cubed gives the volume, which is 27, so 54 divided by 27 equals 2. So as you can see, as you go up this diagram here from smaller to larger organisms, if these represent organisms, your surface area to volume ratio gets smaller. So the bigger we are, the harder it is to get that oxygen. And if you think about it in this cell, it'd be really hard to get the oxygen right to the middle of those cubes, right in the middle of here, into the middle of the volume. The bigger we are, we need that way of adapting to have a bigger surface area. And again, you might be given examples in an exam that looks something like this, okay? So here we've got exactly the same as just a moment ago with you. You've got a cube that's just 1 centimetre, let's say, in length. So it'd be 6 divided by 1. So it would have a surface area to volume ratio of 6. This one, it's got a surface area length, surface side length of 5. So the surface area is going to be 5 times 5, which is 25, times 6, one for each side, which is going to equal 150. And then the 5 times 5 times 5, the 5 cubed to work out the volume, is going to equal 125. So 150 divided by 125 is 1.2. And then the same here for 10. You've got 600, which is the surface area, because you've done 10 times 10, so 10 squared, to work out the area of one side. So you've done 10 there. Just double make sure you're happy where I'm getting these numbers from. Multiplied by 10 there. But there's 6 sides, that only gives you the surface area of one side, so 100 is the surface area of one side. But obviously you've also got 5 other sides, so that will be a total surface area of 600. The volume will be 10 along the bottom, 10 upwards of height, and then another 10 here. So 10 cubed, 10 times 10 times 10, which equals 1,000. So you do 600 divided by 1,000, which gives you a surface area to volume ratio of 0.6. So again, as the objects increase in size, they rapidly decrease in terms of their surface area to volume ratio. And then the question then, why does an amoeba not need to have an exchange system? Well, an amoeba is a single-celled organism. So exactly as we said at the start, that means it's going to have a large surface area to volume ratio, and therefore diffusion is going to easily happen from the environment into the cytoplasm as a result. So when we're talking about good exchange surfaces, then we basically need something that's going to increase the surface area to volume ratio. So anything that's a good exchange surface needs to have a large surface area, and pretty much you're given any example of this, the answers, especially if it's an animal, will always be the same. It doesn't matter if it's a type of animal you've never heard of. So on here, on this diagram, we've got external gills, okay, which are the of the axolotl in this picture. So you can see there that it's got large external gills that would increase the surface area. Other thing you need is a thin barrier to diffusion, because we want to have that ideally 1 cell thick wall effectively, which will allow an increased rate of diffusion, because the bigger the diffusion distance, the greater time it's going to take. So we want to make the diffusion distance as small as possible. We want to maintain a concentration gradient, so that means maintain that higher to lower concentration for simple diffusion to occur over. And the bigger the difference between the two areas, the better. There needs to be a moist surface, because that gives something called surfactant, which helps stops water particles collecting, but it also helps the dissolving of the chemical through the gas through the membrane. We want potentially fluid to carry the nutrients and oxygen around the body, so to carry it away for us, which helps maintain the diffusion gradient. You need a pump inside the body to obviously move things around for you. And you need blood vessels to carry the blood around, and you need 2 circuits. Okay, so when we look then at an example, we need to know about insects, and you're expected to know that insects use the tracheal system of breathing to get the oxygen in. We shouldn't really say breathing, to get oxygen in and carbon dioxide out. And oxygen enters via small holes called spiracles on the side of the insect's body, as you'll see in the next slide. The issue they have, though, is that the spiracles obviously are open and there's an exposure to the body, to the environment. So what happens is we lose water, or insects lose water via the spiracles constantly. So they are adapted to close these spiracles as much as they possibly can to avoid that excess water loss. And there's lots of hairs around the side of the spiracles which will allow water droplets to build up to increases the water gradient between the inside and outside of the insect slightly. So how does it work? Well, basically, if they have valves and tiny hairs, sorry. So how does it work? Well, basically the insects, the oxygen diffuses in through the spiracles into the tubes and then just diffuses down a concentration gradient down the tubes where it splits into tracheoles, and those tracheoles and the oxygen filling up these tracheoles, and then that will diffuse into the cells that it's next to, which will have obviously a lower concentration of oxygen, and it just moves from a higher concentration to lower, and then carbon dioxide just moves back out that way. And they join up particularly at the end with muscle cells where obviously that respiration is happening. Okay, so how does that allow insects to work? Well, obviously smaller insects can actually just do diffusion straight away. More complex insects or more active insects, stuff like locusts in this picture, they might have more holes, obviously more spiracles, but also if you look where they are along the body, they're all along their abdomen. And what they can do is they can contract and expand again like that using muscle contractions. When they do muscle contraction, contracting, it pushes the air out of the body. When they relax their body, and it recoils back outwards, the air gets sort of pushed back in because of air pressure. So again, the insect contracts its body, increasing the pressure inside its body, pushing the air out. And then when the insect retracts, it lowers the pressure in the body below atmospheric pressure, and the air goes back into the insect. Fish then obviously need to do gas exchange as well, particularly important because there's less oxygen available in water than there is in the air for obviously terrestrial insects and terrestrial animals, but fish that are aquatic, they live underwater. They have generally a small surface area to volume ratio. Diffusion would not be fast enough to work. They wouldn't be able to survive. So they need a specialized exchange system that can rapidly get that oxygen out of the water and get carbon dioxide back in. Oxygen is in concentration, so that doesn't help as well. So what fish use is gills, effectively, and the fish have loads and loads of gill lamellae, which are tiny little flaps that the water, as you can see in this top right-hand picture of my head, sort of shows you water can go across these gill flaps called lamellae, and obviously as they are going across it, then oxygen is diffusing into the bloodstream, carbon dioxide diffuses out of the bloodstream. And you can see under a microscope how small and large increased surface area they've got by all the foldings they actually have. Now, fish, there are two methods of gas exchange. And the one that you guys need to be familiar with, but I will describe you about both of them because I think it only helps when you describe both of them, is the countercurrent flow mechanism. So what this means is the blood flows in one direction across the gill lamellae, whereas the water flows in the opposite direction. And if you're ever asked to explain that, even if you can't quite grasp it in your head, what that does is it maintains a concentration gradient. So it means that the diffusion of oxygen into the blood or diffusing carbon dioxide out of the blood can continue along the whole length of the gill lamellae, and it makes sure that there's maximum diffusion of oxygen or carbon dioxide in or out of the gills. Now these two graphs sort of represent this. So if we look at what we call a parallel flow system, which is the second graph actually here, it shows you it's not the most efficient mechanism in the world, because what happens is this. So if we start off with the water, okay, starting off here, the water comes into the gills, and the water has got lots and lots of oxygen as it comes into the gills, okay, which are the two arrows I've shown. But then obviously oxygen starts moving out into the bloodstream of the fish through the gill lamellae, okay, via diffusion. But what will happen, because they're traveling at the same direction as each other, is at some point 50% of the oxygen is in the water still, 50% of it is in the gills. So you will never get this change of 50% efficiency that you get in parallel flow system when the blood's flowing in the same direction through the gills and the water. Now, if you do that opposite though, that changes, and it means using a countercurrent flow system, it's much more efficient. So if we have a look at this, we have the water, which is flowing this way. So you've got high amounts of oxygen in the water here, and the blood is flowing the opposite direction in the water. Now, what that does is it means that once the blood has got to the end of the lamellae, and I should point out that these numbers are incorrect here. Unfortunately, I couldn't get the new picture to copy it, so it should, if we change that to 60 and then to 70, okay. So if we, as they go in opposite directions, what happens is the water, obviously, oxygen starts diffusing out of the water. I'm going to change it up to 40 just to help make sense for you, and it goes into the blood. So at this end, you've got very, very little oxygen in the bloodstream because it's come back from the muscles of the fish, which have been doing all this respiration. So oxygen will diffuse through, and then it will maintain. So there's always a little bit less oxygen in the blood than in the water, so oxygen will move through. And then when we get to this end, by this point, a lot of the oxygen has gone into the blood, so there's quite high levels of oxygen in the blood. But because you're meeting them fresh water that's still got like 100% blood oxygen in, then it's moving across still. So it maintains the diffusion gradient because the oxygen in the water is always a lot higher than the oxygen in the bloodstream. Even once the water is almost passed across the lamellae and we've got low levels of oxygen here, it's still going to be more than in the blood, so it will still diffuse across. So diffusion of oxygen is maintained because of the countercurrent flow system, because there is always more oxygen in the water than there is in the bloodstream, as there is a higher concentration of oxygen in the water than there is in the lower concentration in the bloodstream. The diffusion will always be maintained from a higher concentration to a lower concentration, thus making the system much more efficient and getting more oxygen in the bloodstream for the fish. So I hope it's been useful, guys, and I'll see you again soon.